Rana discourage the use of probability apologetics. Let me get this off the screen. Dr. Rana has discouraged the use of probabilities apologetics, especially to evolutionary biologists. And when this happens, almost certainly there will be a flurry of posts after the lecture of people objecting to Dr. Rana's recommendation from number types just like me. So I'm guilty of that. So this evening, my topic is going to be about protein probability calculations. My presentation will have 3 parts. We're going to do a quick review of protein basics. Then I'm going to present some very typical protein probability calculations, which are prone to imprecision, which I believe leads to the problem of credulity with evolutionary biologists. And then my third part will focus on what is a probability strategy for calculating protein probability. And I have made it a 2-prong, being kind of a hardware, software guy, systems engineer. I like to look at a probability strategy that utilizes the genetic code and its optimization and the improbability of it coming to fruition as a result of random processes. And then I like to focus on the information content, what I call the software that you'll see in protein sequences. So to start, I'm going to go through this fairly rapidly. Protein basics. Proteins require precise 3-dimensional shapes to function in organisms. And of course, the key word there is precise. Precise implies probability, and therein lies somewhat of the debate. Secondly, the shapes are determined by hydrophobic, hydrophilic, and electrical qualities of the surrounding amino acids. And the sequence of the amino acids must be fairly precise in order to affect the exact shape for the protein. Proteins are built from a natural suite of 20 amino acids. To give you some idea of how many different proteins there are in the human body, there are over 20,000 different protein types. The average length in the human body is 480 amino acids, and they all need to be fairly precisely sequenced for shape and chemical function in the human body. Protein folding is a key aspect of calculating information content and probability of proteins. Protein cycle times are also important to really understand the random sequence space that has to be searched in order for a protein to evolve. Times are typically in milliseconds, but the very fastest are in the microseconds, according to Wikipedia, which we all know is the knowledge source of the world. Here's an example of the different structures that you find a protein as output from the ribosome. You have a polypeptide chain. We call that the primary structure. The secondary structure is part of a recurring motif of 2 types of structures called alpha helices and beta strands. And then lastly, from which the real functional protein fold occurs is in the tertiary structure. Now, the tertiary structure, those can also combine in a 4th quaternary structure. And there you see an example of hemoglobin, which is a quaternary protein. Notice how the binding sites are embedded within the quadrants of that globular protein. And the key thing about the protein sequence is those binding sites have to be very close to the physical position within that molecule, or else they won't work. The shape and the physical location are real critical to operation. I'm always fascinated when I look at the beauty and elegance of proteins. They look very elegant, but they also look very fragile. And here's just an example that of cartoons that I grabbed of human proteins. Well, if we're talking about probability, we're obviously going to be concerned with math. And so there is a certain amount of math that we need to be aware of for calculating protein probabilities. And so just as a real quick review, when we see the number 255 in base 10, well, there are other ways to express that same number in other number bases. Base 16, I'm going to be listing powers of 2, which is something that's very, very familiar in computational technology, being powers of 2. So you see the different numerals or the different numbers, different bases for protein probability. We're going to be focusing on the base 4 or quad base. So how do we determine? This is real elementary, but I thought nonetheless I'll mention it. How do we get 255 from base 10? Well, the rightmost digit, 10 to the 0, is the ones, 10 to the 1 is the tens, and 10 to the second is the hundreds. And from that, we get, from those digits going right to left, we get the value 255. So let's look at that. Let's do a calculation of 47 decimal to base 4 quad. And that has some relevance, has a lot of relevance to protein probability. So we work from left to right. Well, 4 to the squared is 16. How many 16s will fit into 47? Well, 32. So that leaves with a remainder 15. So our third digit to the left is 2. And then the remainder, we then take 4 to the 1 into that. It goes 3 times. And then that leaves a remainder of 233, which 233 base 4 equals 47 decimal. So I picked that example on purpose. You'll see here a cartoon of the protein synthesis machine, as I like to refer to it. Messenger RNA comes out of the nucleus. It has a 3-digit base 4 code assigned to it. Transfer RNA has a 3-digit base 4 amino code attached to it. They link together in the ribosome, and a protein polypeptide starting at that singular or the first phase, first structure, we have a protein output from the cell. Base 4 is the coding of the language of protein synthesis. So let's look at the numerals for just a little bit. Nucleobase molecules comprised of 4 nucleobases of RNA. C, G, A, and U, we use the first letter of each one of those nucleobases for the numerals, but we could just as easily assign 1, 2, 3, and 4 to those. In fact, you'll see later on in my research, that's exactly what I did to be able to simulate some of these protein probabilities. I basically assigned C to 1, G to 2, and so forth. So here we have a familiar DNA molecule. We have the RNA molecule, and you'll see how the consecutive segments of 3 nucleobases comprise what is called a codon. And codons all occur in a 3-digit base 4 mathematical encoding. And we've all probably seen this before, but this would be, I lost my place here just for a second. The code definition, you're familiar with the table, which is called the genetic code. I refer to it as the universal genetic code in this presentation. So when you see UGC, that means universal genetic code. The rows in this tabulation, I code as the codon position 3, the columns codon position 2, and with each one of those squares, we see the sequencing of codon position 1. So notice a key thing here is there are 64 codes in the UGC, but there are only 20 amino acids. So obviously there's redundancy or synonyms that are part of the genetic code. Let's look at our number that was part of our refresh exercise. Remember, we converted 47 to base 4. And well, if we take how I've defined 1, 2, and 3, 4, 1, 2, 3, and 4 to C, G, A, and U, that means that we have 233. That equals GAA in the genetic code. And if you target that in the UGC, you'll see that GAA is telling the ribosome that it wants to link the tRNA with something to produce glutamic amino acid. So let's look at some typical probability methods and see perhaps why they are considered imprecise and often rejected by evolutionary biologists. Here's a real common problem. What is the probability of building a small protein, 100 amino acids? That means there's 100 codons. We assume they need to be in a reasonably precise sequence. What are the possibilities? Well, one way you could look at that is these are base 4 codons, meaning there's 3 nucleotides per amino acid. Therefore, 4 to the 300th power gives you a very large number, a number that we really can't comprehend. And I've seen this argument used. In fact, if you look at William Dembski's book, Intelligent Design, that I believe was published in the late 90s, he actually uses this as a method to calculate protein probability. Now, the fact is he's actually correct in that that is the probability of that specific nucleobase sequence, but not of proteins. Why? Because there are only 20 amino acids found in biological systems. So you're exaggerating the probability because you're calculating the probability as if there's 64 unique amino acids. Let's do another method, and you'll see this one more frequently. 100 positions, but we recognize there's only 20 amino acid possibilities, 20 to 100. And there we get a much lower number, about at least 60 orders of magnitude less, a much increased probability, but still a very large number. And one of the things that was made famous in Stephen Meyer's book, Signature of the Cell, is he equated amino acid sequences in protein to a bike lock. So if you can remember when you used to ride a bike to school and you wanted to make sure it didn't get stolen, you would have like a 4-digit byte code, meaning that there were 9,999 combinations required to open that. And so you were counting on a bike thief not having enough patience to go through that sequence space to unlock it. Well, Myers in his book says the probability that we just demonstrated to you with that 20 raised to the power of 100 is like having a bike lock with 131 dials. So if you can imagine the amount of time it would take to go through that search space of combinations, it's an amazingly long time. So what you'll typically find in a protein probability problem is they will give you some idea of just how long that number would take to search through. So remember in our protein basics that the fastest proteins can fold in 1 microsecond. So let's just assume that that's the number we're going to use for the average. If you did that, it would actually take 4 times 10 to the 116 years for that protein to develop randomly if you had to go through the entire search space. And of course, those of us who have had any amount of RTB training know that the consensus age of the Earth is around 4.5 billion years, and the consensus age of the universe is less than 15 billion years. So you're saying, well, wow, protein probability, those numbers are huge. How in the world can evolutionary biologists reject this probability method? And they do. And the reason is, going back to our bike lock 131 dials, because some of the dials actually have more than one number, which will work in the combination. In other words, it doesn't require every site in a protein sequence does not require one unique amino acid. In fact, many amino acids will work at some sites in the protein. So to give you an example of our 4-digit bike lock, let's say, for instance, we had a defective dial, and that defective dial only, it would combine consecutive digits. 1 and 2 would be the same thing. 2 and 3 would be the same thing. So we reduced the number of possible codes on that one defective dial to 5. So when we take 1 over 10 times 1 over 10 times 1 over 10 times 1 over 5, we get 5,000, not 10,000. And therefore, we've increased the probability of opening that lock in a time scale by a factor of 2. And that's a simple way of explaining why many evolutionary biologists and biologists in general reject amino acid or probability arguments for protein because of functional equivalency. So what I'm going to show you on this particular slide are 2 different ways, and I will show a third one later on, of how amino acids are grouped functionally. Here you see charged, polar, amphipathic, which I refer to as hydrophilic, and then hydrophobic. But there's also a second type of grouping, and that grouping is depending on whether you find amino acids highly buried within a protein or whether they tend to be on the surface. So therein lies 2 ways that amino acids are functionally similar, and therein lies why there is some generosity at some site positions of the amino acid that's actually used. So I promised to, my third part of my presentation was to focus on some areas that should be conceded low probability events that occur randomly. And the universal genetic code is most definitely a shining example of something that infers intelligent design or creator. And as a hardware kind of guy, I call it the hardware because it is hard-coded into the chemistry of the molecular machine. The universal genetic code, Crick thought it was a frozen accident. How did this code become fixed as the central dogma of molecular biology? So, I first came upon that term central dogma of molecular biology in my last class with RTB, which was the advanced biochemistry class. And the central dogma of molecular biology was a title given to the protein synthesis machine in the cell. It was given by none less than Francis Crick. And that was, in fact, the title of his paper. And in that paper, Crick emphasized that the central dogma, or the coding of the amino acids that generate a protein function, is not a flow of chemistry or matter, it's a flow of information. And the flow of information is facilitated by a redundant code, that being the universal genetic code. So another person that you will see often quoted in intelligent design lectures is a man by the name of Hubert Yaki. So having studied Hubert Yaki's book, Information Theory, Evolution, and the Origin of Life, it's a painfully difficult book, heavily leveraged in math, but nonetheless, he shows and compares the central dogma of molecular biology as essentially a communication system. And you'll see there that the UGC is actually involved in the encoding and decoding aspect of that biological machine. So let's focus on how well designed that is for protein synthesis and see whether it has inferences and protein probabilities that we can assign to it as to whether or not it actually randomly came to be. Well, something that caught my eye and the attention of all us number guys in the RTB course entitled Evolution versus, actually it was Creation versus Evolution. And one of the things Dr. Rana lectured us about was the error minimization properties of the universal genetic code. So, you know, a Six Sigma guy like me, we salivate when we see bell curves. And because we know when we see bell curves that we can make predictions based on that, which can be very accurate. And it makes a few claims in that slide that was given in that RTB lecture for that class, that the genetic code is 1 in a million. So it's 1 in a million in its ability to minimize the impact of a mutation. So what are the three basic types of DNA mutations? Base substitutions, deletions, and insertions. So let's just look for review. What is a base substitution? I call it a flipping of a nucleotide. You'll see there in our normal DNA sequence, we have a two codons, AGT and CGA. Well, the base substitution in the rightmost codon changes CGA to AGA. And of course, what that does is that actually makes an error in our encoding in the messenger RNA of the amino acid that we want at that particular part of the protein synthesis. The other type is deletion, and this is a type known as a frame shift mutation. So in this example, we're actually deleting the G in the leftmost codon, but frame shift mutations are much more deleterious than flipping of a nucleotide. And the reason is they have a ripple effect, because after they occur, every codon subsequent in the rest of the chain gets impacted with an error. Insertion is also a frame shift mutation, and you can see there the example, the leftmost codon gets a T inserted in the AGT codon, and that has a ripple effect. So in the class, getting back to the creation versus evolution course that we took at RTB, after we saw that slide of the normal distribution and the amazing improbability of the genetic code, 1 in a million per the claim, this number was given 1.4 times 10 to the 70. And it was kind of amazing that all the mathematicians, including myself, in a post thread, we were all trying to figure out how in the world did they arrive at 1.4 times 10 to the 70. And I can tell you, none of us got it right. And of course, we felt like there clearly has to be a mistake because we're too smart to not be able to figure this out. Well, in Yaki's book, he actually does give the equation. And if you crunch those numbers, you will get 1.4 times 10 to the 70. And you'll see there, it's pretty obvious the numerator is essentially 64 factorial for the number of codes. The denominator is a little bit more subtle, but you'll see those factorials, 6 factorial to the cube, 2 factorial to the 9, 4 factorial to the 5, and then there's 3 with only one time do we see that occur. And then there's 2 ones. That actually is the numerator. So what you're seeing in that equation is it's a way to estimate. Actually, it's not even estimate. It's a way to calculate the distribution population of possible genetic codes, assuming that all 64 of those codes can be shuffled. So we're going to get to that a little bit later on. You may have some questions, but let's just have a little bit of review of what normal distribution, sometimes called a bell curve. I have also heard people refer to it as a Gaussian distribution. I've heard these terms used interchangeably, but a normal curve here, I'm showing that 0 is the normalized average. So you get an average. If we want to normalize the distribution, we set the mean to 0. And then along the horizontal axis, we see standard deviations, units of standard deviations, which is also called Z. Some of you have had statistics, have heard the term, what's the Z-score of that sample? Okay, Z can be considered almost the same thing as standard deviation units. Also sigma. So when you hear 6 sigma capability, that's really referring to plus and minus 6 sigma performance for a statistical process. So what can we tell when we see something has a normal curve? What can we tell about that? Well, we can say that we know that if a sample falls within plus or minus 1 standard deviation, that it's within 68% of the entire population around the mean. 2 standard deviations is 95%, and 3 standard deviations is 99.7%. So when we make the claim that the UGC is 1 in a million in error minimization, what we're literally saying is it performs better than 99.999% of all other codes. This is what we call in statistics a left-tailed probability test, because we want to know how many standard deviations does the sample or the number of errors in the UGC have in respect to the rest of the distribution of codes. We call that a Z-score. And if it's less, if on a left-tailed test, if the resulting standard deviation or Z-score is less than our Z metric, then it passes that test. So in the simulations that I'm going to show you for the UGC, we should have a Z-score of less than 4.78 from the entire code population from the mean. I'll give you an example of that when I run this simulation. Hopefully it'll become clear what I'm talking about. Being the geek that I am, I was just fascinated by the amazing precision of the universal genetic code, or at least the claims. And so you said, you know what? I've done a lot of Monte Carlo testing in my career. And so I'm going to put this into my Oracle crystal ball. I'm going to see whether or not it is really 1 in a million. Well, I found out that Oracle did not have a distribution definition that really fit the set of UGC shuffling that I wanted. So I designed my own from an Excel spreadsheet and Visual Basic macros. And so here's the result. Many sheets are part of this Monte Carlo model. You notice in column P, I have listed the different amino acids in their verbose expression. Notice in column O that I've actually encoded the numerals, the nucleobase codes. I've actually translated them to 1, 2, 3, and 4, which is easier for me to calculate. And also in my model, since we're wanting to test, the test statistic is how many errors does the UGC have when impacted by mutations versus the rest of the population. That's what we're testing. So I added an error column and I simulated each one of the mutations types for all 64 of the shuffled codes and compared that to the number of cumulative errors you'll see in the UGC. Here's an example of a couple trials. Once again, the red box around the verbose listing of the amino acids. And trial number 1, you'll see a randomized sample amongst the 64 types. Notice that the equation that I showed you earlier does not assume that the synonyms are blocked in a contiguous block format like the UGC is. And that's kind of a key thing and the beauty of the UGC, which we will see later on. So essentially, my first simulation was, how does the UGC form perform when we have a purely random shuffling without boundaries on the 64 codes? So here is my simulation. The error count for the UGC was 1385. I ran 10,000 trials. And lo and behold, we found the UGC is way left of the distribution, at least on a sample of only 10,000 trials. In fact, it's a z-score of -22. That's well beyond 1 in a million. Now, I realize my statistical sample was only 10,000 out of 1.7 times 10 to the 70th, right? So it's just a small subset. And obviously, the outliers are going to be exaggerated in probability in the test. But nonetheless, the claim held fairly true on my first test. So I want to move on before we do anything more with the UGC. Let's look at information content, which I call the software. And Dembski was famous in his intelligent design book for bringing forward the explanatory filter. And the explanatory filter was a means by which we could detect intelligent design activity from natural law and randomness. That was what it was intentioned to do. And there were 3 conditionals in the flow chart. The first one is contingency. Contingency is is a word that confuses a lot of people. So I like to say contingency basically means, can it be arranged differently? So if we're looking at sequences of amino acids, is there anything in this sequence that mandates that they be in this sequence? Can they be arranged differently? And of course, yes, they can. That's why they're able to be used to synthesize thousands of different proteins. So it passes the contingency test. Yes, they can be arranged differently. Are they complex? And that's kind of to me, the crux of the whole probability issue is, well, the only way to determine that something really is complex is by probability. Now, there are different thresholds of probability. Dembski claimed, well, if something is 1 in 10 to the minus 500, it is impossible for it to happen in nature. And I've seen other examples of the threshold. But the key is some statement or metric of probability needs to be attached before we can say, hey, this thing is contingent and it's complex, but is it specified? And specified is the pattern independent of the event. And for those of us who write software, it's very easy to understand complex specified information. Software is written in a higher language, say for instance, C or Java. We write that. It's a language, a message that's abstracted from the embodiment or the machine that actually executes the code. That's the beauty of it. We don't have to change the code every time the hardware changes or there's an improvement in technology. So software is the epitome of information or CSI. Protein sequences of DNA bases, they're irreducible to the bonding affinities between the bases. So the nucleotide bases can actually attach themselves to the DNA backbone in any order that they want. It's irreducible to a bonding affinity. Yes, they almost all four of them have a bonding affinity to the backbone, but they can take on any sequence. Therefore, they take on a pattern independent of the bonding affinities. So once again, we go back to Yaki's model. Where does the information content of protein synthesis come from? Well, it comes at the very, very front end of the communication system, the information message. And of course, that's encoded and decoded because of the embedded UGC. And it actually results in a targeted function. So what's input on the other side is a specified pattern, pattern to determine a certain function of a protein in the body. Information theory, I would like to say, is another way to calculate protein probability and more accurate. Hubert Yaki, his whole thesis was molecular biology needs to be rescued from a descriptive mode to a quantitative mode. And he was a numbers guy. That's why I kind of liked him. And that's why I got his book. He asserted that measuring information was essential for understanding the nature and origin of life. Now, for those of you who have studied life origins, you know that evolutionary theories are full of just so stories. We could actually call them myths, just like, now myth doesn't mean it's not true. It's just a story that we use to suggest a possible way that something materialized. So Yaki wanted to rescue biology from these just so stories like the primordial pool that life emerged from. I have my own list of favorites. I think every time probability points to intelligent design inferences, a just so story pops up. I'll give examples of convergent evolution, punctuated equilibrium, parallel evolution. I call these just so stories, and to really evaluate them in the spirit of Hubert Yaki, well, let's put numbers to them and measure it that way rather than using descriptions for which opinion is the only thing that matters. So here's a page out of Yaki's book, Information Theory, Evolution and Origins of Life. And we see the key terms in measuring information. P, probability, which of course is the reason for this presentation. N being the length of the protein chain in amino acids. And I being the amino acid in the sequence of which we assign the probability of that amino acid being at that precise location. And that all results in a measurement called information content, called Shannon information. And Claude Shannon, for those of you who haven't heard, he was back in the 1940s. I think he might have won a Nobel Prize for his work in information theory. But nonetheless, it's a log2 encoding of information. So let's look at an example, one that I think you all should be familiar with, because I think this demonstrates how information content is affected by amino acid redundancy. Here's 2 dice that you roll. There are 11 different values, 2 through 12, but notice they have different probabilities associated with the values. One, there's only one opportunity for a value of 2 to come up with 2 dice, right? As compared to 7, the number of values 7, there's 6 opportunities. And notice it has a higher probability. That impacts Shannon content. So the event of rolling 2 dice, that sequence, every event or every site within those events has a Shannon bit value of minus 3.274. So how's that work practically? So we see here, I created a simulation for that. And I just put a random, well, it's not a random sequence. I specified a sequence of 16, n equals 16, of dice values. I started out, went from even, 2, 4, 6, 8, 10, 12. And then I went backwards, 11, 9, 7, 5, 3. And then I worked back up just to fill in the 16. And I came up with a information content for those 16 rolls of 2 dice in that exact sequence of 59.41 in terms of Shannon bit value. That has a probability. Remember, Shannon bits are in log 2. So to convert them to probability, we need to take 2, raise it to the power of the information content, and we get the probability 1.3 times 10 to the minus 18. That means it's going to take you, it's going to be very, 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 very hard, and probably a lifetime you could not roll that particular sequence of 16 with 2 dices. It's very highly improbable. I told you I simulate it. And what do we see? Well, you know what? We can match average. We can match 4 values 45% of the time. That's more than I thought. But when you get down to 16, notice that it exponentially decreases in probability. 1.3 times 10 to the minus 18 was our calculated. Based on the sample of 1,000 that I did, the statistic distribution tells me the probability of that is 2.1 times 10 to the minus 41. So in statistics, when your sample size does impact your accuracy, especially on the outlying scores. So that's why you see the discrepancy. But the closer you get to the mean, the more accurate the predictions. So let's take another example of dice rolls, because I think this one demonstrates exactly what I was saying. Notice that when you roll 2 dice, the middle numbers have the highest probability, with 7 being the peak. In fact, it's symmetrical about 7. 4, 5, 6, 7, 6, 5, 4, 5, 6, 7. Those are the 4 most probabilistic dice rolls that you can have. And when we put that into our pattern, we get a much lower information content, and as a result, a higher probability of occurring. So see the example here? Example 1, where I had a pretty good content of all dice roll values, low probability and high probability, 1.3 times 10 to the minus 18. But number 2, I put high probability numbers, and notice that there is almost a factor of 1,000 difference in the probability. So that is what we're talking about when we are looking at redundancy of amino acids. It's the same effect as some dice rolls have a higher probability than others. Some amino acids, some locations in a protein have higher probabilities than others of coming together randomly. This example, higher probability, well, the average match is 5, and you can see that demonstrated in the graph, and a lower probability. So let's look at information content in closing for protein sequences. Hubert Yaki, his prescription for how evolution brought about all life forms from a universal common ancestor is that it used random mutations acting upon information of a molecule, a protein sequence, and the less information is needed to specify at least one such residue. So because he can prove, supposedly, that there's not a lot of information in protein sequences, therefore it's very, very plausible that evolution randomly created life forms that we know today. Yaki's prescription, it's really twofold. One of them is rather complicated. I call it a physical metric, and it's based on the work of Borsnick and Hofacker from 1985, where they modeled all the 20 amino acids, and they calculated its physical space, where the binding sites might be in a certain location, and they generated a prediction based on functional equivalency because of physical equivalency. And the second one is more understandable by me, and that's a logical metric. In other words, for evolution to work, there needs to be a mutation path from the UGC through a transition of a Hamming distance of equal to 1. So I'm going to explain what Hamming distance is here when I catch up. That Hamming distance means 1-digit difference in a sequence. And to give you an example, in an alphabetic message, let's use my name. My name is Tim. I get confused with Tom all the time. And so, well, that's because the difference between Tim and Tom has a Hamming distance of equal to 1. Mutations generally occur, almost entirely occur, as single point mutations. Therefore, for that mutation to take root in a biological system, it has to have a Hamming distance of 1 in the UGC. I'll show an example later on. So let's take the UGC, and similar to the dice roll, let's calculate the Shannon bit value for proteins, protein sites, general protein sites, or amino acid sites in a protein. You notice here that in the column, my Zoom thing is kind of in the way here, but you can see in the opportunities, you see that column that has 61 in it. Those are opportunities which bias the probability of that amino acid of appearing randomly. The first amino acid you see there, there's only 2 codons out of 61. Therefore, it has a lower probability of occurring, as opposed to leucine, which there are 6 synonyms in the UGC. Therefore, it has a higher probability of occurring randomly. So let's look at, remember early I showed you some typical quick and easy back of the envelope protein calculation, protein probability calculations, 110 amino acid length. I chose 110 because in Yaki's example, he uses 110. So if we were to calculate that in the 20th raised to the length of amino acids, we get a probability of 1.3 times 10 to the minus 143. But if we use the general probability using information content biased by the UGC, we see 1,000,000 times more probability, 1.15 times 10 to the minus 137. So you see how information content is really a much more precise way of presenting protein probability. Here's an example. What Yaki does in his book is he takes the protein iso-1 cytochrome C, and he actually calculates the Shannon bit value of that specific protein for every site. Here's an example of site 16. And this is from, and I looked up the table, his reference material. There indeed is a site on the web, which I have in my references, where you can go to that particular protein and other proteins, by the way, and you can see, wow, site 16 in that 110 amino acid length protein, there are actually 6 amino acids found in nature in eukaryotic organisms, 6 different types of amino acids. So, wow, maybe Yaki is right. And that's, of course, what he's trying to say. His predicted is shown in the bottom. And his prediction is that if the physical and logical criteria that he uses in his prescription, if that's correct, there you will find that evolution will eventually put 13 different amino acids at site 16. So he goes on and he calculates the total information content for the iso-1 cytochrome C. And for the known sequences, what's found in nature right now, 1.5 times 10 to the minus 112. But notice the big difference. He predicts that as evolution winds its way in time, eventually the probability or information content is going to decrease, raising the probability and significantly, as you can see, 30, 40 some orders of magnitude, 6.4 times 10 to the minus 71. So I tried to summarize just the different calculation methods. And you can see as you go from very general, back of the envelope, to very precise as to what's known, which I believe could be very compelling, to those that are predicted, which obviously may not even occur in our lifetimes. But you look at those numbers, you can still see that they're very improbable. In fact, all the sites, some of the sites only have 1 amino acid that will work, known and predicted. So what's my rebuttal to Yaki's prescription? I really listed 6. First one I had mentioned that as even if you take the predicted for a very small protein, 6.4 times 10 to the 71st divided by the number of microseconds in a year still comes out to a search time, if you randomly had to search the entire sequence space, of 2.03 times 10 to the 58 years. That's a long time. It's still a very formidable boundary to search randomly. Also, some of you may know that as you study origins of life, they're doing a top-down origin of life study where they knock out and they try to find what is the minimal number of genes for life to exist. And that minimal boundary is 250. So imagine that the probabilities all concatenate in those 250 proteins, and you get an absurdly large number. So yes, Yaki demonstrates one small protein, and from that he extrapolates all of life. I think that is a major rebuttal to his description. Yaki admits the unknown origin of the UGC very openly in book. He does that many times. And he doesn't really acknowledge where in the world did the information content for the last universal common ancestor come from. In other words, the input to that central dogma of molecular biology, from whence cometh the original information from which the noise and mutation could get creative and generate new organisms. He doesn't really address that. Another one is the Cambrian explosion. You see almost instantaneously in that period a new appearance of information with new cell types. New cell types require new proteins. Here's an example of the phyla cell types and organ grade types from the Cambrian explosion. A massive amount of information has to be generated, and very low probability, very high information content. Another thing that is I believe, a very solid rebuttal is research. Back in the 90s, an MIT molecular biologist by the name of Sauer, he determined the ratio of functional to non-functional sequences to be 1/10⁻90 for his testing with 100 amino acid linked chains. In 2006, another paper was published in the Journal of Molecular Biology, which estimated that 1/10⁷ sequences are capable of function or actually folding into structures that could possibly be functions. So, and that goes down a rabbit trail I don't want to go, but the function actually is even a lower probability. And lastly, of my 6 is the existence of transcription factor proteins that regulate gene expression networks for the cell. So early in my electrical engineering career, being the low Sparky on the totem pole, I had to do wire apps of very complex designs to test them out. Of course, nowadays you just simulate it. You don't have to go through this exercise. But back in the old days, you populate the one side with what I would analogize to functional genes. But on the other side, you see the complexity of the wiring. And that wiring, I think, is very analogous to what transcription factors do. Regulatory genes help express the functional genes. Yaki admits and acknowledges that there are several ways that nature has incorporated redundance for error correction in the genetic code and in the protein message. Notice how he capitalizes N, and he does this throughout the book. It's almost as if he is making nature a god. And so my suggestion is, if you could take Yaki's argument and suggest perhaps that capital N is the God of Scripture, because I believe a strong case can be made for intelligent design. 2 papers that are often cited in our RTB classes, the genetic code is 1 in a million. I'm sure that's where they got that slide claim. Notice the highlighted sentence. It has been shown that the code minimizes the effects of point mutation or mistranslation. Erroneous codons are either synonymous or code for an amino acid with chemical properties very similar to those of the one that would have been present had the error not occurred. That's kind of a key statement. And just a year or so later, another research team came out with more research, and they said, you know, it's not 1 in a million, it's actually 1 in a billion. And notice that last sentence highlighted in blue. When alternative codes are restricted to those that interchange biosynthetically related amino acid, the genetic code appears even more optimal, 3 orders of magnitude more optimal. Here's the thing that caught my eye when I read that paper. They did an analysis in this later research for fitness of function in the effect of a mutation. And they found that if the mutation resulted in an equivalent amino acid amongst these 4 different groups assigned here, that there was still a high level of fitness resulting from that mutation. And that's how they made their research determine that the UGC was very effective. So I said, I wonder if that's true. So I decided to go to my simulator and factor that in. But first of all, I generated a Hamming code matrix. Along the top, you'll see the amino acids on the horizontal axis, and along the left, you'll see the amino acids along the vertical. And everywhere you see a letter in that matrix, there is a Hamming distance of 1 with that particular, between those 2 amino acids on the 2 axes. So I know that's really hard to see. So I wanted to zoom in on just a zone of that matrix. And lo and behold, what you find are 3 examples of shared functional biosynthetic groups. In other words, Y in the red box, Y, F, and S are all part of the biosynthetic pathway shared. So if a mutation occurs, because Hamming distance equal 1, mutations are typically single point, it's typically going to mutate to a functionally equivalent amino acid. Likewise for the other 2. L, H, Q are all part of the same biosynthetic pathway categorization. I, M, T, N, and Q, there you see just in that small zone, a shared linkage of functional equivalency between that group, Hamming distance equal of 1. So I decided, and that's what I'm going to do in closing. I said, you know what? I'm going to simulate just what the UGC code looks like statistically if we assume biosynthetic pathways with a Hamming distance equal of 1. Well, if we do that, I decided, well, it's well known that the contiguous block nature of the UGC is very important for error minimization. So I changed my equation to only use a population defined by the denominator. And when you do that, there's about 522.5 billion codes possible. And here's the model. You'll kind of see this from the examples here in the red boxes. The UGC assignments, notice that the synonyms all are consecutive in blocks. So I changed my Monte Carlo model to make them all be randomized in their original contiguous blocks to see whether or not the UGC actually performed better in that way than a randomized contiguous block. So you'll see 2 trials, assignment N plus 1. Remember, I did 1,000. Actually, I did 100,000, but I wasn't going to show that statistic tonight. So let's see how it did. Well, first of all, when we just simply look at contiguous blocks, obviously the error code of the UGC doesn't change. It's still 1385. Let's do our 10, 100, 1,000, 5,000, and 10,000. And wow, all of a sudden we find out the UGC is still better than most, but it's more closer to the mean in just a distribution of 1,000 trials. It almost looks more like that graphic that I saw in the creation versus evolution class. So it's kind of more what I expected to see. But remember, I'm modeling just that denominator of the contiguous blocks of the UGC code. What are the statistics? Well, the UGC scores a minus 2.9 or 1 in 90,000 if you simply look at it like that. So I decided, okay, I'm going to put the biosynthetically equivalent amino acids and group them together. And I'm going to look at an error, and then I'm going to analyze it to see whether or not it mutates to a biosynthetic equivalent amino acid. And there you see the column highlighted in red. I did that for single point, nucleotide, and insertions and deletions. And well, immediately the thing that came out is, wow, the cumulative error code of the UGC was nearly cut in half. It went from 1385 to 733. Remember, Yaki said it's the UGC has an amazing fault tolerance associated with it. This statistic definitely demonstrates that. We almost cut in half the number of serious deleterious effects by mutations. Well, how does it do compared to the rest of the distribution? Once again, I go through the trials, 10 through 10,000, and lo and behold, we find out that the UGC shines brightly once again as a major outlier to randomized contiguous code sets. I can't see what the mean. I guess the mean is about 1173. The zoom thing is right in front of that graphic. But what is the Z-score? All of a sudden, the UGC uniqueness is brought about and amplified. It scores a minus 8.2, which has a left tail probability of 9.4 times 10 to the 17th, as compared to contiguous contiguous genetic code sets, which we know will yield better error minimization. So I know I've gone through this very quickly. I have 2 pages of references that I'm perfectly willing to share. I'm not sure what your protocol is for doing that, but that's the end of my presentation.
Thank you, Tim. A little bit over some of our heads, I think, but I think you did a good presentation. Why don't we open it now for any questions that people have? So if you want to ask a question, unmute yourself and ask your question.
I'd like to ask a question for anybody that can answer it. When you genetically modify a protein, like with the spike protein, in the mRNA vaccines, for it to insert into the ACE2 better, and also to give it a bigger pathway. What other influences does that change have on the protein itself? Like does it remain stable, or can it shift it, or like I'm very interested in, for anybody that can give me an answer to that.
Well, I'm not a biologist. I'm more of a mathematical guy in probability, but what I've researched on that is it is very likely that there will be a change. But the key thing is that will that change destroy functionality? And therein lies, remember, Yaki used these models of physical placement. He looked at how did the different angles in that part of the protein change, and do those changes in angles make the function go away, or do they make that change toxic, or do they maintain some fitness of function? I'm assuming if you're talking about the COVID vaccine, that they've used these very same techniques to determine just what happens when that particular area is mutated. Does it maintain function? But I'm no biologist. That's just coming from how I would apply some of the information that I've learned.
Okay.
I have a quick question for you, Tim. I've been doing a lot of reading over the years because I'm a biochemist, and I've actually done a number of presentations on origin of life, and I talk about the genetic code. So two things really. The first one is, has anybody looked at the first level of epigenetics in terms of the control of the genome at the universal genetic code level? And specifically, I'm referring to the fact that there's only one codon that is the start codon, which causes the start of translation from RNA to protein. And that's the same codon as codes for methionine, as I'm sure you know. What is the probability that any code would emerge randomly that would have a single codon that drives all translation activity?
Yeah, and I appreciate your comment, because I agree with you. It seems highly improbable that would occur. And I really do believe, and I've read about epigenetics and its role. So when we start, that's a whole nother discussion about how epigenetics and the probabilities of how it interacts with the nucleotide basis in expression. I guess the point of my, Don, you're far more qualified than I to answer that question. But for me, as a numbers guy, I think it is it's not very courageous for us as apologists to concede the probability argument. Mm-hmm. And that therein lies my passion. And maybe it's ignorance is bliss, that maybe I don't know enough biology to understand that no, these things can randomly come together and function. My interpretation, remember what we're told in 1 Peter 3:15. Everyone should give a reason for why they believe. Now, many of us take proxies, right? Many of us, some people take Ken Ham as a proxy. Whatever he says is what I believe. Or some of us take Hugh Ross as a proxy. Some of us take Francis Collins. Well, yeah, that's all fine and good, but each one of us has an expertise that we need to diligently find reasons for why we believe what we believe. My expertise is statistics, which deals with the probability of events occurring. That's why I'm very passionate about it. So when we concede protein probability, and you mentioned the fact that I can't even pronounce that methylene, whatever that start amino acid is, it seems absolutely absurdly improbable that people would think that code could randomly appear. Francis Crick, he couldn't understand it. Yaki says it cannot be known. It's just something we'll have to live with.
Thanks, Tim.
I have one question. I still don't understand really why the evolutionary biologists reject the probability. The only reason I've heard is that they assume that you don't need to make the whole protein to start with. You can make parts of it that do other things and then combine those parts to make a bigger protein. Have you heard of that?
Yeah, I think that's what Yaki's argument is that some of these amino acids will coalesce into sequences and they will combine with other sequences, and the probability of them arriving at a functional sequence is actually much lower than people think. That's his thesis. And he explains that in his book. What I find interesting, and for those of you who have taken RTB courses, Hubert Yaki is quoted frequently. In fact, the quotes typically exhibit a very intelligent design friendly quote. And they support high improbability, the high improbability of these happening by random. And so when you read Yaki's book, you think, wow, I'm going to be hearing a lot of good stories about intelligent design. The fact of the matter is, Hubert Yaki is an ardent Darwinist. And in fact, he essentially ridicules irreducible complexity as a valuable argument. His answer to that is, well, the DNA molecule eliminates irreducible complexity as an argument because it defines how these things are built. You don't need to have one part all functional before the other parts work. So he denounces irreducible complexity all within about 3 or 4 sentences. He really doesn't do it justice, but he also says there is no need for an intelligent designer. So he really ridicules a lot of some of the 3 pillars that Dr. Rana recommends: irreducible complexity, number 2, biological systems, the analogy to machines, and then thirdly is the empirical. Yaki would take the first 2 and said, no, amino acids can kind of coalesce and create functionality. And the whole reason we know that happened is because we're here. Now, I call that a just so story, which of course he denounces as a descriptive and not quantitative. So it's really interesting to see what he calls implausible, because I believe his whole comprehensive argument is implausible, but yet he holds to that. And if you read his book, he's an amazingly smart man. The math in this book is well above my pay grade and most people's pay grade. It took me a long time to really understand how he calculated information content. So, but yeah, Bruno, I think you've got it. Because functional equivalency coalesce, they can coalesce randomly and they get together as a team and voila, you have protein functionality. And then when you get 250 of those, says the top-down origin of life theorist, all of a sudden you have life. And by the way, Yaki admits that this all happened in 200 million years, just after the late bombardment. And it's just, to me, it is validation of C.S. Lewis when he said, you're never going to change a heart with an apologetic. You can remove obstacles of the mind, but you can't change the heart. So Yaki holds firmly to his materialist worldview, and nothing is going to shake from him. That only comes through the power of the Holy Spirit.
Yeah.
Tim, sorry, I have another quick question. Can you comment just briefly on the concept of parity coding? Because I've read a number of papers on that.
Yeah. Well, I'm not, the parity coding is mentioned a few times. Case in point, it was actually controversial in one of the classes I had with Helmer. And Dr. Rana spends several pages describing how the nucleobases have a parity code that's used for air checking that the bases don't mistakenly combine, and it uses a parity code. So Bruno, are you familiar with what a parity code is?
Yeah, I'm a software developer myself.
Okay, so when I read, and Helmer agreed with me, by the way. I love Helmer, by the way. That guy is a brilliant man, and when he writes a post, you can tell he's thought about it very, very well, and he researched real well. And if he is typical of people in your group, you have a great group of people, and you have a great ministry. But nonetheless, he and I both agreed that Dr. Rana's explanation of parity as it's used in the connection of nucleobases in the DNA molecule is not really a very good parity argument. Helmer and I both agreed to that. So I'm not a big believer in the parity code as it's described in the DNA molecule. Now, perhaps you're referring to parity in some other biological system, but the only one that I've heard described is the one in the DNA molecule, and I think that's a bad analogy.
Thanks, Tim. That's why I asked you, because it seems like it was popular a while ago, but it's fallen out of favor, and I just wanted to hear you confirm that. So thanks.
Well, you know, and I think Helmer and I had several exchanges about this. It seems that it's more a physical or mechanical check than a parity check. So it's not within the spirit of what parity is all about. Right. And for those of us that are familiar with parity and how parity is used with Hamming codes and error correction, it's just, I think, not a very good demonstration of parity. And it kind of really, I think, weakens all of his other arguments. And I think he may think that that's a great argument, but then again, he's a biologist. He's not a Sparky or a computer guy.
Anyone else have a question?
I could probably look this up myself, but while you're here, the Cambrian explosion, just what level of complexity of life arrived during that period? Was it just the things that you showed us? Excuse me.
In that particular slide, that was taken out of Stephen Meyer's book, Darwin's Doubt. He devotes a whole chapter to the number of phyla that actually appeared in the Cambrian explosion, essentially in I think 10 to 50 million years. And that explosion of information content, there is no way that comports to Hubert Yaki's information content erosion in that short a time period. So with all those phyla, and I don't have the time to get that book for you, but that chart that I give you is a demonstration of some of the, let me see if I can get to it. It's a demonstration of some of the cells that had to occur. Whoops, there we go. So there are some of the phyla identified that occurred. If you look at the fossil record, they just appeared. Okay, the inference is those phyla are very complex, and they're complex in very different ways. They have different cell types. Some of those cell types are organ grade, which means they're very, very different. They have a lot of information content. From whence do they come? And to me, it's the same if you're a materialist or naturalist, it's the same problem you have with the origin of the universal genetic code. It just happens so quickly that it's unknowable. Now, if you read more of Meyer's book and how he describes the argument from paleontologists who are of the evolutionary paradigm, they suggest that the ancestors to the Cambrian explosion phyla were all soft-shelled, and therefore they were not capable of having residue fossils. That's why we're not finding them. So I look at that as a just so story. Maybe it's right, but it doesn't seem right, because the hard shells appearing in the phyla at the Cambrian explosion definitely require new types of cells, which is new information. Yep. Where does that come from? How could that have evolved in such a short time period? I don't know if that answers your question, but Myers does a real good job of explaining that in Darwin's Doubt. In fact, I would say a major part of his thesis is the questions that arise as a result of finds in the fossil record, and namely the Cambrian explosion sediment.
Yeah, I appreciate your answer, and that complexity arises suddenly, which does suggest something. But my question was simpler than that.
Oh, I'm sorry. You know, my wife says that I don't know how to answer any question simply. So that's case in point. She'll get a little laugh after about this question when I debrief with her.
That page is showing 7 different phyla, but there were actually 70.
Yeah, I'm just giving you an example. Yeah.
But the point is that 70 phyla all coming into existence in a geologic, instantaneously, an instantaneous moment of geological time. It begs the question, how? And I loved your answer about the hard shells, because it's still, okay, fine, let's concede that that's true. You still got to explain how the hard shells all came into existence, all in a split second, relatively speaking, of geologic time. So no matter what you do, wherever you turn, you have to explain why the sudden appearance of information and complexity.
Okay, accepting the sudden appearance and all that. My question is more like, I know the whales came along at a certain period and the dinosaurs came along at a certain period. Was this the level of complexity, these things here at the Cambrian explosion? Was there anything more complex than that, or was that much later or a little later?
The idea here, Andrew, is body types. These are all the different body types that have ever existed. So if you look at the different ones that he's showing, they're radically different in body type. So we share the same body type with mammals, but also with other animals as well. We're a tube within a tube, basically, which is a single phyla. It's a type of phylal structure, but there were over 70 different ones of those. And so you're not talking about animals that are different from each other, but fall into the same phyla. We're talking about different phyla, which is different types of body types. Mm-hmm. That's what's radical about the Cambrian explosion.
Okay, all right.
Tim,
maybe Tim can do some math on that for us.
Yeah, I'll set up a simulation tomorrow and give you an
answer.
Right.
Absolutely. Yeah.
Tim,
you'll need a few supercomputers.
When you heard Dr. Rana explain that these sorts of arguments don't convince the evolutionary biologists, did he relate any stories of conversations he's had with evolutionary biologists and what sort of arguments they use against probability?
Well, first of all, I love Dr. Rana. Obviously, I've read every one of the books that he's ever written, probably. So I'm grateful for his ministry and his work. But he's a biologist. I'm a numbers guy. And And I kind of took that advice to mean, you know what? I am never going to be able to convince an evolutionary biologist with an argument about biology. I'm just not up to their pay grade. He is, therefore I have to respect his experience in doing that. The answer that I remember him giving, and it was a short one, was we just don't understand enough about proteins to make a probability claim.
Oh, okay.
Yeah, you got to wonder if he's on to something or if he just had a bad day, got discouraged.
No, he's not just had a bad day, Andrew, because he's actually mentioned that in no less 2 different lectures in 2 different classes. So, I take it as that, in fact, the one where he makes the strongest recommendation about that is in the evangelism to science class. I don't know if any of you have taken that, but in there, they have training on effective apologetics for physicists, chemists, biologists, geologists, and all the different types. And they bring in an expert of that particular science to describe apologetics that are familiar. So I think of Dr. Rana's recommendation as being in the context of arguments to evolutionary biologists. And he's saying, don't go there because you're not going to get any traction. I think that's what his recommendation is. So I think he's being very specific to that discipline.
But your arguments may convince somebody with a mathematical background.
Exactly. Like, I would like to have a discussion with Hubert Yaki about his book, because I've read it, from cover to cover and every which way in between. It's highlighted, and through all the typos and everything else. Unfortunately, he passed away a few years ago, but his daughter has maintained a website where she does entertain questions. And so I did send a question to that website, and it never was returned, never replied. And that website, interestingly enough, Hubert Yaki's daughter is heavily involved in the LGBTQ community and the politics associated with that, but not the liberal politics. It's the conservative politics. And her claim is that there is an extreme fringe of the LGBTQ community that's really making a bad name for the rest of us who are very reasonable, quiet people, not demanding a lot of rights that other people don't have. So I would say she's as interesting as her father, but nonetheless, her passion doesn't seem to be information theory.
Tim, you mentioned that currently we don't know enough about proteins. Does that mean that, let's say I wanted to make a protein to do a certain function, is there enough knowledge to make a brand new protein that would do a specific function?
Yeah, I believe, and I'd have go look at some of my books in the RTB courses, that yes, they've synthesized proteins and to do different functions. Yes, they have targeted different functions. I can get back with you if it's really important to you, but in I believe it's in Dr. Rana. He wrote a book, Creating Life in the Lab. I don't know if any of you have read that title. In that, he has a section on laboratory synthesized proteins for new functions. And it's somewhat controversial because it's kind of like they're playing God. And what if they create this protein that eats the rest of us or something like that? So I believe you'll find a good answer to that in Rana's book, Creating Life in the Lab.
Okay, thank you. I have that book. I haven't gotten around to reading it.
The critical feature of that book is that although it's possible to create probably synthetic life at some point, at least a simple, fairly simple working cell, the bottom line is that it requires a phenomenal amount of intelligence and circumstances to be absolutely perfect as set by the experimenter in order for any of it to happen. And so the more complex, the greater the number of conditions that have to be perfect. And that's the whole point of the book, is that when we look at the real world, it just begs the question of, you know, there must be a designer.
Well, thank you, Tim. Maybe I think we'll end it here now.
I got to ask one last question to the rest of you about Toronto. I worked on an integrated circuit design in Toronto many years ago, and I did that in the dead of winter. And I was told that Toronto is the tropical zone of central Canada. And of course, I did not find the temperatures to reflect that categorization while I was there. But is that a true statement, that Toronto is relatively mild compared to the rest of central Canada?
Yeah, we're near the Great Lakes, so
yeah,
might be.
Okay.
Yeah.
We're not really.
So I guess I'll need to come back there more.
We're not central Canada because that would refer to like the prairies.
Okay. Well, they said they were the tropical zone. I thought they said of Canada, but I thought, well, what about British Columbia and Vancouver? I thought, well, that's pretty.
Well, that's a lot milder than what Toronto.
Yeah, absolutely. So when they made that comment, I thought, well, that sounds like quite an exaggeration, but nonetheless, thanks for answering. So I just want to thank you all for allowing me to share tonight. And when Hilmer gets back from the Ukraine, let him know that I appreciated him thinking about me and my passion to give me an audience with you guys. I appreciate it very much. And I'm grateful for the opportunity and your time.
Well, thank you for joining us. This has been recorded, so Hilmer is going to watch the recording. And you can probably contact him in a few weeks to give you his opinion.
Okay, I will do that.
Thank you.
Well, bye you all.
Bye everyone.
Thank you, Tim. Good night, everybody.
Good night.